later. To kick this off, I will start. Give me 1 second. So, as I was saying, I, oop, 1 second. I'm a senior research fellow at the Center for Enzyme Innovation. Um, Hopefully most of you have heard of us. Over the last few years, we've done a number of sort of big profile publications and work towards understanding and developing enzymes for the biodegradation of plastics. The focus on my talk is basically introducing the group here, which is obviously filled with diverse chemists, to enzyme catalysis for plastic degradation, walk you through what the center does in terms of what our team looks like, and then show you an example of both some science from our group, some recent completed science from our group, and also how we look forward at the work with enzymes. So most of you at this point are familiar with the structure of PET, which is a homopolymer of repeating TPA ethylene glycol units. It was discovered in 2016 that an organism, Idenella sacchiensis, expresses two enzymes that allow for the enzymatic breakdown of PET to constituent monomers. In fact, the monomers used to produce PET to begin with. This inspired a great deal of efforts across the world in pursuing additional enzymes that might actually break this down a little bit more efficiently. It's not that Idenella pettase was the first one identified, it's that the organism had had the potential to actually metabolize the product. So it was able, in principle, to digest this enzyme or digest the plastic down into basically fuel for itself. It turns out that that's probably not the case, but in essence, what the organism was doing was essentially what we want to do. So that is to digest and break down PET plastics down to its constituent monomers for upcycling or recycling back to PET. Because in principle, once we break them down to the monomers, we can actually re-engage the plastic completely back to PET as if it were virgin PET from petroleum resources, or we can use these products to actually
Apologies to
grow.
interrupt, Bruce. Can you hear me okay?
Yes.
Your slides are not moving forwards, unfortunately. I think you might maybe be sharing the wrong part of your
Okay.
screen.
Sorry
Or
about
not
that.
in present mode. No problem. Just thought I'd mention it now.
um, do you see that now?
So it's moved on to the second slide.
Yeah.
It's not in presenter mode yet, but I can see
Does
that it's
this
gone on to the second slide.
in the third one now?
Not yet.
Okay, that's weird.
Have you pressed the presenter mode in the bottom right-hand corner?
I have. So let me try
You
this
have.
again. Yeah. I can try this. Let me try that. Is that working better?
Um,
Excuse me. You have to change the monitors.
Okay.
You need the main monitor for or the primary. I think that one. Yes.
Yeah, let's try
That
that.
might work.
How's that? Is that working?
Not yet.
no.
So maybe the other one.
Sorry about this, guys. How about that? No?
Also
No
no.
progress.
Perhaps just go in and out of it and see. What...
1 second. Let me try to convert this to a PDF.
Yeah, either that or completely stop sharing and try sharing again and just the whole screen.
So yeah, 1 second. Okay, so entire screen. I'll do this, share, and let's try that one more time. Can you see that?
Yes,
good.
useful.
And this, it's working?
Yeah, it's working.
Okay, good. Sorry.
Awesome.
Enzymes are great, basically. Thank you, guys. Because again, most of us probably in this talk are not biochemists, I wanted to briefly walk us through what the enzyme is doing at the surface, because it is somewhat unique. And so we can envision this sort of soluble dynamic protein, which then subsequently interacts with the surface in a sort of bound enzyme substrate complex. It then proceeds through a tetrahedral intermediate, forming our first oxyanion during the reaction progress. And then in principle, it could actually go one of two ways. It can either stay attached to the plastic and complete its chemistry cycle on the plastic surface itself, or it can be released from the plastic and continue the hydrolysis in solution. Which cycle it chooses actually depends quite a bit on what ester bond it's breaking within the plastic. And it is clear from the product distribution that we see that actually both of these pathways are functioning. What's important here to realize is that there are quite a few steps that are underway, and that engineering this enzyme, in fact, engineering any enzyme with this sort of complexity of reaction cycle is quite difficult. The most important feature is actually knowing what the rate limiting step is. And so the rate limiting step, like all enzymatic or serine esterases, is that you need this sort of water step. It's the second hydrolysis, it's the hydrolysis step where water is introduced, activated by the enzyme and allowing for product release. Despite the fact that we know this, very few steps have been taken to rationally engineer the enzymes with this knowledge in place. In fact, most of the engineering to this point has been somewhat obscured by just sort of random mutations that have been identified, or in principle increasing the structural stability. I'll talk about some of those efforts in a bit. But we've actually learned more about how these enzymes work by studying a broad family of them. And so for those of you in the field and for those of you outside of the field, we've actually identified a number of enzymes. It's not as many as you might expect. I think the latest number is around 30 something confirmed enzymes that work on plastic itself rather than a model substrate. But what we can learn from these enzymes is that they have elevated activities at higher temperatures, so at temperatures close to the polymer glass transition temperature. And you can think of this as basically making the substrate more appropriate for enzymes to act at it, rather than perhaps accelerating the enzymatic mechanisms. The other things that I think chemists really want to think about, particularly when we're dealing with this sort of heterogeneous catalysis, where the catalyst is coming from solution down, is that the rate limiting step isn't this attachment, and it doesn't seem that the binding of that enzyme to the surface is kinetically limiting. We see saturation kinetics, as we'll discuss in a bit, at relatively low concentrations. The enzymes have a nanomolar affinity to the surfaces and can be washed off quite easily. Some of the other developments in studying broad classes of these enzymes is that their active sites are important in terms of the dynamics of them. Basically, more rigidly active sites, the worse the enzymes perform. And this can be thought of as a consequence of where these enzymes are originally coming from, and also the consequence of the fact that the plastic itself is heterogeneous. So most of these enzymes are actually cutaneases, hence the CUT name that you see repeated over the class of enzymes here, this family tree. And these cutaneases actually cut cutin, which is not a homogeneous polymer. It is a sort of random smattering of ester bonds that plants produce to produce their outer bark, etc. And so these enzymes are naturally capable of binding to and interacting and hydrolyzing a range of different substrate geometries. And that flexibility is likely what's imparting their activity on the plastics as well. On the other hand, this flexibility, as we'll discuss later in this talk, has some negative effects that we have to really think about when engineering and thinking about these things. And then finally, one of those sort of worst features of the enzymes is that plastics are complicated. They have multiple different solid phases within them. So there's an amorphous phase, a sort of more rigid amorphous phase, and then a crystalline phase. These enzymes do not work on crystalline PET. And unfortunately for us, most of the PET that we interact with on a day-to-day basis, these clear bottles for colas or the clamshells for food, are crystalline PET because they have very nice optical properties at that level. And this means fundamentally that at least at the current state of engineering, these enzymes really have very little, if any, activity on crystalline PET. This is not to say that engineering hasn't been quite successful. There's been quite a bit of a number of approaches taken towards improving the enzyme's activities across a range of different approaches. Here's a smattering from the last couple of years. In two cases, people are using machine learning in an iterative fashion here to improve the TM of the enzyme, so the melting temperature of the enzyme. And then in this report, actually using machine learning model that's been trained on enzymes in general to identify point mutations that may accelerate this. It's a really remarkable paper in terms of the simplicity of the mutations introduced and the rate enhancement that was observed. And then finally, I want to highlight, because Tony is on the Net Zero Committee, work from Tony's lab where through a directed evolution approach, they were able to actually enhance the activity of idenelipetase through a series of laboratory evolutionary steps to get it to be quite robust. In fact, I think under some conditions, it's even better than the reported best enzyme prior to this in the literature. And so with that being said, it feels like the community is quite full of engineers, and so what is it that the center, where I'm a senior research fellow, does that might be slightly different and might allow us to really sort of push the limits here. And so I want to talk briefly about what we're doing at the Center for Enzyme Innovation in terms of our structure, in terms of our integration across a broad swath of scientists, doing everything from digging for new organisms in the muds off of coastal regions to industrial or pilot scale industrial applications of these enzymes. And so the Center for Enzyme Innovation is separated into several groupings. This is the discovery team is pretty much what you'd expect of it. These are scientists who are interested in identifying new patases or even how patase activities enhance the function of the organisms that they are within. That information is fed back to the engineering team, which focuses on improving both characterizing the enzymes itself, but also improving their functions through both rational, semi-rational, and laboratory evolution techniques. And that information sort of flows back and forth between the teams, because as we discover new insights into how these enzymes are working and what rate enhancements, what mutations allow for certain rate enhancements, we can feed that information back into the discovery team to help guide their searches. And then sort of more critically to this, and this is one of the things that's sort of missing in larger scale efforts to pursue these things, is that we actually have two additional teams that are composed of members of the center. And one of them is the deploy team, where we take these enzymes that we're working in lab and we scale them up, right? Our goal is to really be able to use these for an industrial process where we're actually using the enzymes to process the plastics down to its monomers to allow us to reuse these things. And to do this, we really need to build into our pipeline an approach that allows us to do these reactions at scale. So we have several different bioreactors which allows us to digest plastics at elevated volumes, but also elevated solids loadings, which is relevant for industrial processes. And then finally, a relatively recent addition to the center since I began, is the idea that we can actually then take these products of the enzymatic reaction and complete that cycle, right? Our goal is to form a circular economy of the plastics, at least in part. And so we break them down, we purify them, and repolymerize them. And this really does allow us to look forward in terms of what is needed to allow us to address the problems of plastic pollution, indeed address the usability of plastics going forward. And really, the fact that this is all in single center allows us to have this sort of deep engagement at different levels, because to be able to apply these enzymes really robustly, we need to have engineered them with that in mind. And so we're doing this as a sort of singular pipeline. We're a well integrated group, and I want to introduce just briefly the researchers doing this work. And so we are a group of 9 or 10 research groups that all fall within the center. All the groups here have projects that are not simply plastics, but we have come together to form the center. We have our microbiologists and our marine scientists who are really looking for new organisms out there to produce and degrade plastics. Sam Robson's been doing some fabulous work with COVID recently, but is also using the same sort of techniques to understand and sequence sequence the transcriptome and genetics of organisms that might have plastic degrading enzymes within them. The engineering team is sort of a core component of this, and is where I'm centrally located. And we have a number of biophysicists and structural biologists and molecular dynamics experts who are really looking at improving the quality of these enzymes. So really understanding their function, and then taking that knowledge to the laboratory and improving their quality. And then finally, and not least of which, we have the deploy and apply team, where, as I said in the previous slide, our goal is to really understand these plastic surfaces, what's happening at the surface, and then sort of regenerate plastics or whatever from the materials that we generate by their digestion. And then finally, most importantly, is all the people who are actually doing the work in the labs, our PhD students and postdocs. And so what is it that we do? And so this is sort of a broad outline of broad slide of some of the recent results that we've been looking at in the team, everything from identifying enzymes in sort of an expanded class of using bioinformatics, so driven by members of my team, but also by Sam Robson's team, sort of exploring the sequence space, using that expanded sequence space to sort of engineer new enzymes, in this case, using developing enzymes on the basis of their sort of bacterial ones, feeding that information into molecular dynamic simulation and structural biology to help us do structure guided and rational engineering of these enzymes to improve their function. And then last and not least is exploring how the enzymes are actually changing the surface of the plastics and ultimately how they're functioning at industrial like scale, so at very high solids loadings. And so what I want to talk about today, I'm going to really focus down on a story where we're sort of integrating all of these sort of concepts together to try to solve one of the sort of underlying mysteries of the plastic degrading enzymes. And then I'll walk us through some recent life cycle analysis from our team and our collaborators at NREL. So when you're thinking about catalysts at surfaces, particularly ones that have a volume or size, you can start to imagine that you have this sort of solid plastic and the enzyme is sitting down, right? The enzyme has some dimensions to it. And that at some point in time, that plastic surface is going to be saturated and you're not gonna be able to get any more additional enzymes. And you're gonna have sufficient amounts of enzymes to coat the surface. And in fact, that's sort of the optimal scenario. This is the sort of saturation model for processing. So this is a inverse Michaelis-Menten plot. So what we're seeing is essentially a rate of product formation versus enzyme concentration as opposed to substrate concentration. And we see saturation behavior not only for the total, but also for the individual products that are being produced. On the other hand, if you thought about it a little bit further, there's also another possibility where that first enzyme sitting down might sit down in a way that blocks other sites from occurring. And you begin to think that, okay, there's a possibility that you might actually crowd out enzymes. At very high protein concentrations, you may actually start to see an inhibition. The problem with the sort of simple model that we sort of all come up with when we're sort of building this is that idea of blocking other sites is not mathematically accessible. So it doesn't actually yield the observed result. What this type of behavior requires actually is that the enzymes are interacting with each other in a semi-specific manner on the surface. I love throwing in the pictures of the coupons just showing how much changes occur during the processing, just to give people really some insight into what's happening. So when thinking about this problem of this autoinhibition, basically, because it's an undesirable feature, because we want to be able to use high enzyme amounts to drive reactions to completion, and if we're sort of killing our activity, but by having too much enzyme, it's really something we don't want. And so if we want to approach this sort of rationally, which is understand where this is coming from, and allow us to basically prescribe a way to avoid it through engineering approaches, but hopefully in the process of doing this, understand what's fundamentally occurring. And so one of the sort of key insights into what is fundamentally happening under all of this is that the products of the reaction have different extents of inhibition. What I mean by this is that if we imagine that our inhibition is measured as a ratio of the activity of the enzyme producing, say, mHet at 500 nanomolar versus its maximal activity, we see that the mHet is completely killed. That activity of producing it is completely killed at 500 nanomolar, and TPA, for instance, is not completely killed. And this should be shocking to all of us because it doesn't fit with sort of product inhibition concepts, which is what we're sort of introduced to. It's actually rather looking more like a substrate specific autoinhibitory behavior by the enzyme. And so we begin by thinking about this question. So why are these two lines different? Why do they not match up? And it really quite quickly dawned on us that the terminal ends that the plastic presents to the solution are what matters here. To make TPA, iodinylpetase and other petases can only cleave the very terminal piece of the plastic that has an acid group. Basically, only is basically generating a diacid, and therefore the terminal group has to be this carboxylic acid. Um, in contrast, there are multiple different ways of forming MHET. You can cut both ethylene glycol terminated plastics, but also the acid terminated plastics. And so, what we think this is sort of suggesting to us, or at least in that initial result evaluation, was that how the enzymes are interacting with the plastic surface varies based upon the ends of the plastic. And this makes some intuitive sense, right? The enzymes are going to have preference for binding with one type of plastic over the other. By their very nature, they're going to be somewhat fickle. And so we began to realize that this wasn't a sort of non-specific phenomena, but rather that the enzymes are interacting with the plastic termini, especially in terms of catalytic cycle, very differently between them. And so we wanted to really understand this subject a bit further. And so we began to look across a broad range of what are called type 2 petases. And these are the mesophilic enzymes. So iodinylpetase is a sort of founding member of this group, and to be honest, these are the least likely to yield industrial process at this point because they're not naturally thermostable. On the other hand, understanding how and why they have autoinhibitory behavior is important for us to understand what's happening at the surface. And so by exploring this relatively large group of enzymes, some of which are new to the literature, we actually got really lucky. There's an enzyme actually relatively closely related to petase, so RGCUT2, that actually has no autoinhibitory behavior at all. This was a shocking result because we were hoping to be able to just write that this is a characteristic of type 2 enzymes, but having discovered a type 2 enzyme that doesn't have it, we suddenly had the ability to use structure and sequence-based insights to really explain this phenomena. And so when we we did quite a bit of work, but to sum up as quickly as possible, when we began to look at the sequences and structures of these things, we noticed that there were sequences that were different around the active site. This is not surprising; enzymes do vary, but most critically, we observed that there was a change of an alanine residue to a phenylalanine. And that is a weird mutation because there are substantially differences in sizes between these two residues. And indeed, looking at voids near the active site, so this is the active site histidine of the catalytic triad, we see that a void behind the active site loop in iodinylpetase is actually gone from RGCUT2. For most of the biochemists here, this would immediately suggest changes in dynamics, and I will get to that in a moment. But for the others who maybe aren't so used to looking at and thinking about protein structures, basically you can envision this void as something the protein wants to avoid. And so there's going to be a lot of dynamics to sort of partially fill that void over time. So the time integral of the kinetic or the dynamic behavior will attempt to close this void as much as possible. There may be even water incursion occasionally. And so what this immediately suggested to us was that we could actually generate a mutant of the RGCUT2 to reintroduce this void to see if that actually causes it to suddenly become autoinhibitory. And lo and behold, by reintroducing that void using a fairly conservative set of mutations, we were able to see that that enzyme actually becomes autoinhibitory again. It's not a substantial amount of autoinhibition, but it was enough for us to begin to realize that this is probably what's causing these issues. And so we then subsequently went through the literature, both from sequences that our team has produced and also sequences from Anthony Green's group, and we identified that actually through directed evolution, hot petase, one of Anthony Green's enzymes, loses its active site dynamics. It actually becomes much more sort of frozen in place, which is what you would expect when you fill this void. And the fact that this is correlated with loss of autoinhibition gives us a rough model for what we think is happening here. And that is basically that the enzyme can bind to the plastic in several different ways, some of which are productive and some of which are not productive, depending upon the dynamics of the site. And under crowded conditions on the surface of a plastic film, that this will have the tendency of pushing the enzyme into a non-productive conformation, and hence you see autoinhibition. It's a rather simple model. Obviously, it requires further confirmation with surface level structural studies. These are well beyond basically anybody's expertise in the world at this moment, and so we are pushing out this work as sort of a descriptive model, and hopefully we'll follow this up with additional studies going forward. And so what does this study sort of show us is that, well, despite all of the engineering that we've pushed through in the last few years across the world on these enzymes, we really don't understand how they're working. Despite this, their industrial prospects are quite high. And so for this last bit of the talk, for the last few minutes, I want to introduce where enzymes are in terms of industrial processes for recycling PET, and where we can go in terms of improving things going forward. And so this is from a life cycle analysis paper published by our collaborators at NREL and John McGeehan. And so this is sort of what the process would look like. The enzymes are here. And then everything else around this is about recovering of products on this side and repolymerization. And everything prior to that is basically on pre-treating of the plastics as necessary for getting the best operating enzymes to work. And so I want to walk through, this slide is very busy, but I want to walk through briefly where enzyme engineering can actually come into play to decrease the cost and effort required here. Before I go into that, I think because this will probably come up again in this seminar today, is that there's quite a bit of use of sodium hydroxide and sulfuric acid. There's this attempt to do recrystallization of the products. And so we're dealing with common problems that exist both for chemical recycling efforts and biological. So when we begin to think about how we can move enzymes forward, the most fundamental challenge for us is actually getting those enzymes to work on that crystalline PET phase. The reason for this is that there's quite a bit of energy investment needed to take what is crystalline PET and turn it into a amorphous powder that the enzymes like. This is possibly it requires quite a bit of insight and quite a bit of work going forward. These enzymes simply don't like crystalline PET. It's a very well ordered surface, probably not incredibly dynamic. But we do have some insights into how to drive these things apart, whether it's chemical treatments or finding new types of enzymes that might be able to impart some energy into that surface. We're hopeful that either pre-treatment processes or identifying a different sorts of enzymes may actually yield a reduction of the cost here. And this is really great because what this means is that if we can get rid of this process, then we can get rid of quite a bit of this process as well, this sort of cleaning and washing of the plastics, because the enzymes, one of their advantages is that they can deal with dirty systems, right? This is they're evolved in a complex biochemical milieu, and the fact that you have to clean them and make these sort of pristine substrates is not a product of the enzyme, rather it's a product of the chemistry to get to that point. Furthermore, although it might not seem so evident, changing the enzymes or engineering the enzymes is also quite helpful for reducing downstream effects. In essence, what we want to do is reduce the amount of volume that we're doing these reactions in. So we need high solids loadings, and in fact, some of the most likely scenarios for getting this to work is actually running the enzymes in what are called moist conditions. This is like barely any solvent, where essentially TPA itself will crystallize out, which means you don't have to add sodium hydroxide in the reaction itself, which means you also don't have to use sulfuric acid and sodium hydroxide in the subsequent reaction. And it also makes, for instance, purifying ethylene glycol even easier. And so that sort of approach where we're looking at trying to put enzymes into sort of this for lack of a better term, amorphous mush to do the reaction rather than in stirred reactors is really something where enzyme engineering can move forward. And then on that point, our enzymes are pretty good. So a lot of the best enzymes out there, including hot petase and the LCC variants that Carbios has generated, already promote greater than 90% depolymerization under high solids loading. In principle, though, we can do better, right? So even at 97 point something percent, we're still going to be pushing solids through to the next stages. As I just mentioned, we really want that chemistry to work at low volumes. And so engineering these enzymes to work at sort of somewhat moist conditions is always sort of useful. The other side of this is part of the study of improving these enzymes is also understanding how the biological systems outside of the laboratory are going to be dealing with these things. And so if we can really push the engineering and the evolution of these things in laboratories going forward, we're going to better understand how biology response. This sort of summarizes basically everything I've just said. There's really a need for the enzymes that work on crystalline PET. A lot of the engineered enzymes don't actually last very long, so that requires reintegration, reintroduction of the enzymes for long processing. And so what we really want is faster, long-lasting enzymes that actually function at lower temperatures. So if we can get this chemistry to work at below the TG, then we're going to be able to actually have enzymes that have a less intensive process during the bioprocessing side of things. The other side of this is extending the pH tolerance of the enzymes. This only gets you so much. It's actually much less than you would think. There isn't that much sodium hydroxide needed over the course of the reaction. It's sort of equimolar to the product of the reaction, so lower pH doesn't get you very much. It's essentially the same calculation. But more importantly, things that I didn't talk about is eliminating product inhibition. This is something that all enzymes and sort of technological processes have to deal with at some level. And then we're really interested in applying the lessons learned to other polymers. So understanding how the soluble dynamic enzyme is working and functioning at a surface is really is the insights that we have, we believe, can be applied to other polymers, not just condensation polymers like PET. And then finally, looking forward, it feels like a lot of the work that we're doing and that the field is doing in general is that we're chasing plastics that were not conceived of to be sustainable. And we're doing this because they work very well. But what we probably really need is actually approaches where we're generating plastics that are optimized for the enzymes or optimized for a sustainable practice, rather than chasing what the ghosts of the 20th century have produced. And so one of the things that allows that we're excited about in the center is that we have this integrated approach, and that we might be able to begin to pursue engineering polymers that are basically specified for the enzymes that we're developing. And with that, hopefully then coupling those two products together for basically a sustainable future. And with that, I would like to thank everybody at the Center for Enzyme Innovation, especially the folks that worked on the project that I talked about: Luzana, Liliana, Gerhard, Rosie, and Professor Pickford. And then, of course, our collaborators at NREL. I'm going to plug myself, my group, just briefly. We do have postdoc and PhD positions available, and you can find my email address there. Cheers.